Good afternoon, students. Good afternoon. Whether I'm audible? Okay, welcome you all. Okay, in the morning section, we have completed first 60 question of previous year 2025 plant biotechnology portion. Okay, now we are going to see the remaining 60 questions. Okay, be attentive in class. If you have any doubt, you can ask me there itself. Okay, remaining 60 questions we are going to complete in this session. Okay, with this we will complete our previous year question paper for 2025 plant biotechnology. Okay, first question, simple thing only. Okay, first question, which among the following ribosome is present in the bacteria? They are asking about ribosomes that are present in the bacteria. Any guesses, student? Ribosome present in bacteria. Excellent. 70 years. Okay. We know in prokaryotics like bacteria, ribosome will be 70 years. In eukaryotics, that will be what? 8 years will be there. This will be having two different subunits. One will be the largest subunit, okay, 50 years, and smaller will be 30 years. This can be 60 and 40 years. So, the right option for the ribosome present in bacteria is 70 years ribosome. Okay. Yes, clear. Indian government has allowed export of onion to 6 countries despite ban on shipment. Okay. The thing is, they are asking, identify the state which is the largest exporter of onion. Okay. Which is the state? Largest exporter. Excellent. Maharashtra. Nasik market. It's one of the Asia's biggest market for onion export. Okay. So, Maharashtra is the largest state that is producing onion and exporter of onion from India. Okay. So, the answer is Maharashtra. Okay. Third thing, observe the question properly. They are asking here what? Control of gene expression, particularly in eukaryotics. Okay. They are asking about at which levels the gene expression will be controlled in eukaryotics. Okay. What will be the answer? Yes, whether only at the transcription level, or in transcription as well as epigenetic level, or it will be there at epigenetic, transcription and translation level. Yes, excellent. All the above. In eukaryotics, there is a multiple level regulation will be there. Okay. It can be regulated at transcription level, post-transcription level, translational, post-translational, as well as at epigenetic level. So, this will be the right option. Okay. Okay, clear. Now we are going to next question. Okay. Simple question they are asking about mitosis and meiosis, and a simple definition they are asking, that two direct questions. See here, nuclear division followed by cytoplasmic division. They are saying nuclear division is followed by cytoplasmic division. This is also known as what? Yes, excellent. It is also known as karyokinase. Okay. So, nuclear division is happening followed by cytoplasmic division. This is a general term known as what? Karyokinase. Then what is mean by cytokinase? What is mean by budding? What is mean by cell division? See, cell division means the pre-existing cell will be divided into different daughter cells. Okay, general term. Okay. Budding means what? This will be very common in yeast. What it will do? Here one organism will be there. This will be budded. This budded form will produce into a new organism. Cytokinase is what? Simple. Another form of cell division. So, the right option, the nuclear division followed by cytoplasmic division, is also known as karyokinase. Okay. Next question. Our favorite topic also, nitrogen metabolism. Okay. They are asking about denitrification. Okay. The denitrification is a multi-step dissimilation nitrate reduction process having great signification in sewage water treatment. This is clear. The sequential order from first to last in which of the following nitrogen compound appears as an intermediate in this process? This we already discussed in our class. Okay, what will be the correct answer? We know denitrification means what? Conversion of nitrate to nitrogen gas. Okay, so the first product will be what? First substrate is what? Nitrate, and the last product forming is what? Nitrogen gas. They are asking here first to last. So first will always come as nitrate. Second, then last product will be nitrogen gas. We can go by elimination method whether nitrogen gas is there. Yes, dinitrogen is there. So this will be the last. So D should become as last. So this two option will come. Okay. If you see the normal reaction, how it will happen? Nitrate will be there. That will be converted into nitrite. Nitrate will be converted into nitric oxide. Nitric oxide will convert to nitrous oxide. This will be converted into nitrogen gas. Clear? Nitrate, nitrite, nitric oxide, then nitrous oxide. At last, nitrogen gas will be removed. So, the correct option is what? Yes, nitrite, nitric oxide, nitric oxide, then dinitrogen. So, option A is the correct option. Excellent. Option A is the correct answer. The nutrient element that is involved in stomatal regulation. What is the nutrient element involved in stomatal regulation? The opening and closing of stomata. Excellent. Potassium. Potassium is the nutrient element involved in opening and closing of stomato. Which is the hormone involved in stomatal regulation? Plant hormone. Which is the plant hormone involved in stomatal regulation? Which will be the hormone? Auxin or cytokine? Excellent. Abscisic acid is the hormone involved in stomatal regulation. Yes, very good. Very good. Very good. Next. Which membrane have greater binding capacity than nitrocellulose membrane? Think the question. They are asking about membranes that are used in what? SDS. They are used in, what are the membranes they are used in? We can use nitrocellulose membrane or nylon membrane. These are the different membranes that are used in blotting techniques. Okay. They are asking in this question which have the greater affinity. The greater affinity will be there for what? Nylon. Excellent. Nylon is having the greater affinity. Yes, correct, Uday Lakshmi. Next, they are asking here general thing, goods buffer. Okay, the name of the person is goods. That's why they are saying goods buffer. He has classified this buffer into different categories. Whatever the buffer that is having a capability to maintain the physiological pH of 6 to 8. Okay, that buffers are coming under the category of goods buffer. Okay, based on certain characters he have classified. Okay, so they have given here the list of buffer. They are asking in this question which is not falls under the category of goods buffer. Okay, which is not falling under the category of goods buffer. Any guesses? See, first Hep's buffer, MOPS, these two are commonly used. This two will come, this will have an physiological pH range of 6 to 8 that can maintain the biological significance of the biomolecules. Okay. Trypsin is what? Trisglycine. This is also another common buffer they are using. So the right option is what? Tris buffer. Tris buffer is not coming under the good buffer classification. But trisglycine will come. Trisglycine is different. Tris is different. So the right option is tris buffer. I hope this is clear. Okay. Another thing they have asked from translation point of view. They are asking about genetic codon. Okay, which is not true for genetic codon. Okay. Genetic codon means what? It's a triplet of nucleotides. Triplet of nucleotides that code for specific amino acid. Okay, specific amino acid will be code. Okay, it will have certain characters. Okay, first thing, a codon is a triplet of nucleotide. So this will be right. Second thing, what they are saying, during translation, the codons are read in a non-overlapping fashion. This is also true. Yes, this is also true. Then the amino acid sequence of a protein is linear sequence of contiguous triplets. This is also true. This we have seen. But you see the last option, in a given single stranded DNA or mRNA, all possible reading frames liking to encode a given protein. This is wrong because why? If you see the sequence of amino like mRNA, there will be a nucleotides, for example, adenine, guanine, thymine, cytosine, like this nucleotides will be there. Okay. There will be three possible reading frames will be there. Okay. First reading frame can start from here. It will read the codon like this. Then second reading frame start from this guanine. This is reading frame 2. Then third can start from this cytosine. This is reading frame 3. Okay. The protein synthesis from each and every reading frame will be differ. Okay. It will not be same. So the wrong option about the genetic codon is option D in this question. Okay. Yes. Next, in plant tissue culture, they are asking about plant tissue culture. Okay. Cell viability test. How they are doing the cell viability test? Okay. Among the cell viability test, which cannot be used to determine the viability of the test. Okay. They are asking which test we will not use among this test for checking the viability of test. Okay. See here. First, TTC, tetrazolium, yes, we will use. FDA, this also we can use. FDA staining we will use. Yes, Gus will not come. Yeah. Staining with Evans blue, this we have to learn. Okay. Evans blue, what it will do means the speciality of this Evans blue is it will stain the dead cells. Okay. Living cells will not be stained. Clear? So by differentiating this one, we can check the viability of the cell. Okay. Gus is what? It's a reporter gene that we cannot use for checking the viability of the tissue. Okay. Clear. Next. Yeah, we already morning itself we have shown. TK, now price has been dropped. TK only 2000 rupees per the course. TK Plant Biotechnology course. You can join in that. Then exclusively mock tests are there in our mock test bundle course. This also you can join. Then those who want to prepare for next year JRF examination, our exclusively course is available from now onwards at the rate of 4999. You can join in that. TK. Next question. The main advantage of CAM metabolism. What is the main advantage of CAM metabolism? Any guesses? What will be the main advantage of CAM metabolism? Whether nitrogen fixation will be there, reduced light requirement will be there. Yes. Water conservation in arid environment. Okay, this is the most advantage of CAM plant. Okay, how it will conserve the water? By diurnal. Diurnal means what? Day and night time, the stomatal opening will be varied. Okay, the stomata will be open at night time. Okay, and closed at daytime to prevent the water loss from transpiration. By this process, it will efficiently conserve the water. Okay, that is the CAM metabolism advantage. Okay. Next, identify the microbodies present in the plant cell. They are asking what are the microbodies present in the plant cell. Okay. Microbodies. What will be the microbodies? Yeah. What will be the microbodies present here? This is a common and simple question from cell organelles. This we have already seen. Peroxisome and glyoxysome are the most important microbodies. Okay, microbodies means peroxisome and glyoxysome. Excellent. B and D is the right option. Next, This is very important question. Observe properly. Calculation. Okay. They are asking the relationship between percentage of absorbance and transmittance. Okay. This question is from what? Spectrophotoscopy. They are asking spectrophotometry. Okay. They are asking the absorbance and transmittance are measured commonly in spectrophotometry. This is clear. Yes. Absorbance can be calculated from the percentage transmission. Absorbance can be calculated from percent transmitted. How to calculate? Using which of the formula? TK, they have given different formula. How we can calculate the percent absorbance? Excellent. Can come, money can come. Excellent. TK. We will see the calculation now. TK. Absorbance formula is what? Minus log T. TK transmittance. This is the formula. TK. But if you see in this option, they have given in percentage of transmittance. TK. Percentage of transmittance means what? It is T by T into 100. Okay. If we want to substitute the value for the transmittance, so T equal to what? Percent T by 100. This is clear. T equal to percent T by 100. Just I am substituting the value here. Log 10 percent T by 100. Okay. I am just doing log m by n. Okay. Minus log 10 percent T minus log 10 100. Log 10 100 value is what? 2. If I simplify these things, it will come 2 minus log percentage of T. Clear? This is another important derivation. They can ask based on this one question they can ask. Okay, so the right option is absorbance equal to 2 minus log percentage of T. Okay. Here they are asking about how the electron transport chain happening in photosynthesis. Okay, light and light reaction, how it is happening, from where to where electron transfer is happening. This question is very simple one. The order of appearance, first to last of the following electron carriers in Z scheme. They are asking about Z scheme. Okay. Photosynthesis representing the path of photosynthetic electron flow from water to NADPH. What will be the path? We know here PS2 will be there. This electron will be accepted first by which component? Yeah, CBDA. Okay. This will be accepted by pheophytin. From here, plastoquinone will come, cytochrome B6F will be there, plastocyanin will be there, PS1 will be there. Here, what? Here different things will be there. Iron sulfur cluster will be there, ferredoxin will be there. This is the general pathway. Okay, general pathway. But they are asking, see the option here. They are asking from one to last how it will go. Okay. Okay, from here to here how it will go. They are asking. Okay. So first thing, it will always start with PS2. Okay, this PS2 here only what? Oxygen evolving complex will be there. Okay, so the first thing will come manganese containing oxygen evolving complex. So first will come C. Okay. Then from here where it is going? It is going to plastoquinone. So second will come plastoquinone. Okay, from plastoquinone where it is going? It is going to plastocyanin. So this will come at third step. And the fourth step, what it will come? It will come under P700. Clear? This is clear. So the right option is what? Option B, CBDA. Okay. Next question. Identify the short day plant. This is simple thing. This is simple and direct question. What are the short day plants? Okay. Example of short day plants. Any guesses? Any guesses? What will be the example of short day plants? Yes, soybean is an example of short day plant. Any other guesses? Yes, soybean. Then option A. Yes, soybean and cotton is an typical example of short day plant. Okay, this is direct question. Okay, they can ask this one. Okay, any other example? Yes, tobacco is also an example of short day plant. Then apart from that, chrysanthemum also will come. This is also an example of short day plants. Okay, you just remember what are the example of short day plants, long day plants, day neutral plants, and short long day plants and long short day plants. Okay, so total 5 different classification is there. Based on the photoperiodism, they have classified. You just go and remember the examples. Okay, example for short day, long day, day neutral, short long day and long short day. Okay. In which region in YAC vector, East artificial chromosome, the new DNA is introduced in the cloning experiment? Yes, option D. Option D. Suppressor 4. Any other guesses? Suppressor 4. Why? Why suppressor 4? Yes, suppressor 4 is the right option. See here, for example, this is the vector. Okay, this is the YAC vector. For example, this is the region that will code for SUP4. What it will do means, it's a tRNA suppressor. tRNA suppressor. Okay. If we clone our target gene in this region, this SUP4 will be mutated. If this is mutated means the colonies will be white color. Okay, colonies will be white color. So the region where they are using is SUP4. Okay. Screening how they will do means white and red screening. White, red screening, not blue screening. White and red screening. Okay. Recombinant will be white, non-recombinant will be red color. Okay. Okay. Someone is suggesting, guys, read Instant Note Plant Biotechnology, everything in it. Okay. Someone is suggesting the book also. Okay. Already we have discussed A to Z in our course itself. You just go through our course material, that is highly sufficient to crack this exam. Okay. We have compiled the contents from standard books, Leninger, Instant Notes, everything we have given there. Okay. So you blindly you can follow our class notes, that is highly sufficient. Okay. Next thing, the host yeast strain. Okay. YAC vector is a auxotropic mutant for which of the following gene? Auxotropic mutant for which of the following gene? Yes, tryptophan auxotropic mutant will be there. Apart from that, what? Uracil synthesis enzyme. This tube will be mutated in that strains. Okay, this is the right option. This is very simple question also. Okay, next question. The breakdown product of DNA identified as a plant hormone in cytokinin, as a cytokinin. Which is the right answer? Which will be the right answer? This we have discussed in our class. Yes. The breakdown product of DNA identified as a plant hormone cytokinin is, yes, yes, kinetin. This we have already seen. It's a synthetic cytokinin. This we have already seen. Trans-yet in what? It's a natural cytokinin. This also we have seen. This directly came from our lecture only. Okay. Which of the following is true for PBr322? This also we have discussed. PBr322, plasmid BR322. It contains only ampicillin resistant, or it contains both ampicillin and tetracycline resistant gene, or the cloning site is present only in the ampicillin resistant gene, or it is a natural vector. What will be the right option? Whether it will have only single selection marker, single antibiotic selection? Yes, it will have both ampicillin and tetracycline resistant gene. This we have already discussed. So the gene can be cloned, the gene of interest can be cloned either in the ampicillin resistant region or tetracycline resistant region. This we have already discussed. Okay. Which is the following is not a software for dot plot analysis? Okay. From bioinformatics, they will ask 2 to 3 questions. Okay. This you have to just read like continuously. Okay. What are the softwares? Major software used for major techniques. Okay. So here Laligan is not a software for dot plot analysis. Remaining everything they will use for this dot plot analysis. Okay. You just have to remember these things. Okay. Next thing about nitrogen metabolism only they are asking. Observe properly here. They are asking which of the following NIF gene, nitrogen fixing gene, is most commonly used as a molecular marker for identification of nitrogen fixing microorganism. Okay, nitrogen fixing microorganism. What will be the right option? What will be the right option? We know this nitrogenous enzyme will have what? Two component will be there. Component 1 and component 2 will be there. Okay, component 1 will be exclusively made up of what? Alpha 2 submit will be there. Okay, then that will be coded by NIF, DK and NIF H. You see in this option, the major NIF nitrogen fixing gene they have given in the option is NIF H only. So that will be the exact answer for that question. Okay, that will use as a marker enzyme. Okay. Another question they have asked about DNA replication and its enzyme. Okay, DNA replication enzyme they have asked. Arthur Karenberg and colleagues purified and characterized a DNA polymerase from E. coli cell. Okay, who has discovered this enzyme? This is true. Arthur Karenberg only discovered. Okay, from E. coli, this is known as DNA polymerase 1. In our class itself, we have discussed this DNA polymerase 1 have different functions. Three major functions will be there. One is what? 5' to 3' polymerase activity will be there. 5' to 3' exonuclease activity will be there. Then 3' to 5' exonuclease activity will be there. We can also say this as what? It's a proofreading function. Proofreading 1. Okay. This is what? Primer removal. So these are the different functions. The most important thing is all these function will be present in a single polypeptide. It's a single polypeptide enzyme. N-terminal to C-terminal will be there. Here, if we do proteolysis, hydrolysis of this fragment, it will be cleaved to two fragments. One fragment will have the two functions, 5' to 3' polymerase activity and 3' to 5' exonuclease activity. And the second fragment will have 5' to 3' exonuclease activity. I hope this is clear. Okay. You see here, the second option they have given, E. coli DNA polymerase 1 is a multi-subunit protein. This is ultimately wrong. It's a single polypeptide enzyme. So the right option is what? Statement 1 is correct and statement 2 is false. Okay. Clear? It's a single polypeptide enzyme. If we do proteolytic analysis, like proteolytic cleavage, that can be give two fragments. These fragments have different functions. Okay. Which of the following vitamins was discovered first? Which of the following vitamin was discovered first? We know A, D, E, K. This they have named based on the discovery only. TK. First discovered is vitamin A. TK. TK. Clear? Then B complex vitamins will be there. You have to remember what are the different names of B complex vitamins. This is also another important point. TK. From vitamins part, you will get one question. Okay, you have to remember what are the B complex vitamins and what are the different names and what are the major function, how it is acting as a cofactor for various enzymatic reactions. Okay. Next, the Ca-rich sequence called RUT element is associated with, any guesses, students? The Ca-rich sequence called the RUT element is associated with, whether it will associated with transcription or translation, initiation or termination. Transcription initiation or transcription termination? Translation initiation or translation termination? Yes, it's a transcription termination process. Then most important thing, that is particularly in prokaryotics. Okay. Prokaryotics. Okay. The transcription termination in prokaryotics will be happen by two ways. One is row dependent transcription termination. Second is row independent. Okay, row independent termination. Okay, the simple thing is in row independent termination, there will be a sequences that will be self-complementary to each other. Because of that, a transcription termination will be happen. In row dependent termination, there will be a one protein, okay, that is RUT. This will have a CA rich sequence that will lead to synthesis of AU rich sequence, okay. This will form a complementary things. Helicase will come there. It will terminate the transcription. You see here, this is the thing, okay. RUT protein will be there. Here CA rich will be there. This is just an helicase protein, okay. So it will remove the synthesis mRNA from here. So transcription will be inhibited. This is particular for prokaryotics, okay. That is also another important question. How many structural genes are present in lac operon? How many structural genes will be there? This is simple question. Lac operon, how many genes will be there? 1 or 2 or 3 or 5? We know that lac operon, okay. Lac operon arrangement, if you see, means there will be a 3 structural genes will be there. Yes, 3 structural genes will be there. Lac Z, Y and A. Okay. This will have a promoter region. Okay. So this will have a promoter controlling this LAC, Z, Y and A. Okay. Z, Y. This will code for beta galactosidase. Okay. This code for beta galactosidase permease. This will code for transacetylase. Okay, simple. See, operon means what? A single promoter controlling the expression of several genes. See here, a single promoter is there. It is controlling the expression of catabolic genes involved in catabolism of lactose. Okay. So there are 3 structural genes are there in lac operon. Okay. So the right option is what? Structural gene is 3. Okay. 3 structural gene will be there. Clear? Structural gene only 3 will be there, not 5. 3 structural genes will be there. LAC, Z, Y and A. Okay, clear? Next, ubiquitin is related to, for what purpose? Ubiquitination will be happen for what purpose? Whether for protein synthesis or degradation, or protein binding or protein DNA binding. What will be the right option? Ubiquitination is happening for which purpose? Yes. Excellent. Protein degradation. You know, this ubiquitination, what? It's a protein. It's a 76 amino acid protein. Okay. This is a ubiquitin molecule. Just imagine. This is a ubiquitin molecule. Okay. It will have glycine as 76th amino acid. Okay. What it will do means, this will go and bind with the target protein. Okay. For example, some protein is there. Okay. This will form a isopeptide bond. This amino acid will form a isopeptide bond with the target protein. This will be targeted for 26S proteasome. Okay. 26S proteasome. Nothing. This protein will be completely degraded. If a protein is binded with the ubiquitin moiety, so protein degradation will happen. Yes, clear. Next, which of the following nucleases has a FOC1 restriction enzyme domain? Any guesses? FOC1 restriction enzyme domain will be present in which? Zinc finger, RISC, CRISPR or DICER? This we have discussed in our lecture series itself. Yes, zinc finger nucleus will have FOC1 endonuclease domain. What is the other genome editing technique that will have this FOC1? What is the another editing technique that will have this FOC1? Any guesses? Yes, talents. Talents also will have FOC1. Talents. Yes, excellent. Talents. This both will have zinc finger nuclease domain. CRISPR will have Cas9 domain. This we already know. CRISPR will have a Cas9 domain. Okay. Then they are asking about the function of ethylene. how ethylene is involved in flowering. See, although ethylene inhibits flowering in many crops, it induces flowering in some of the crops. What are the crops? Where ethylene is inducing the flowering. This I have told in our plant hormone class itself. Yeah. Any guesses? Any guesses? Yes. If you see our lecture notes, it's there. Yeah. Excellent. A and D. In mango, for synchronizing flowering, they will use ethylene. Then specifically in pineapple, ethylene will be used for inducing flowering. So, A and D will be the right option. Excellent students, excellent. Next, simple definition about plant growth. They have asked, okay, the ratio of amount of water lost through transpiration by a plant and the amount of carbon dioxide assimilated. See here, it is releasing the water, okay, and uptaking some of the carbon dioxide. What will be the process? Whether that will be a transpiration ratio or water use efficiency, quantum efficiency or carbon use efficiency. It's just releasing the water molecule. Yes, water use efficiency. Okay, any other guesses? Transpiration ratio means what then? Then what is meant by transpiration ratio? Hmm? What is meant by quantum efficiency? What is meant by carbon use efficiency? You just go and see what is the exact definition of water use efficiency. The right option for this is transpiration efficiency. Okay, transpiration ratio. I told here clearly, they are asking if water molecule is going out and CO2 is coming in. This is what? This is nothing but it's a transpiration ratio. How much of water is going out and how much of the carbon dioxide is assimilated. So the right option is what? Transpiration ratio is the right option. Okay. This is another important thing they have asked from enzymology, but little much difficult question they have asked. Okay. These are some of the deciding factors that will decide the top 5 ranks. Okay. They are asking here mechanism of each and every enzyme. Okay. Whether it's an induced fixed model, how the catalytic activity is there, how the activity is regulated, like that they have given here. Okay. You observe here clearly, if you see here, they have given 4 enzymes: triose phosphate isomerase, hexokinase, aldolase and enolase. If you see, all these enzymes are coming in glycolysis pathway. Okay, clear. We know each and every step of this reaction also we know. Okay. You see, observe here properly, lead to synthesis of high energy compound capable of driving ATP synthesis. If you see in glycolysis, okay, substrate level phosphorylation will happen in 2 places. One at last step, the conversion of phosphoenol pyruvate to pyruvate. There ATP will be synthesis. Okay. Then another thing is what? Conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. There one ATP will be formed. Okay, substrate level phosphorylation will form. So they are asking here, lead to synthesis of high energy compound. That reaction will be catalyzed by what? Enolase. Correct. Enolase will catalyze that reaction. So D will come option 1. Where is coming? D is option 1. So this 2 will come option 1. Okay. Then the most important thing is, if you see the action of aldolase, what that will do? It will convert fructose 6,1,6 bisphosphate, to glyceraldehyde, D-glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This stereospecificity will be more for this enzyme. Okay. You see here, where is, so stereospecificity is more for which enzyme? Aldolase. So second option will come C. Here only it's coming C, second option C. In both option it's coming second option C. Okay, second option will be C. Then hexokinase. Hexokinase, what it will do? Hexokinase is the major enzyme. It where the substrate glucose will bind, then water molecule also will be added there. That will lead to conformation changes in the enzyme activity. Okay, conformation changes in the enzyme activity. So for B, 3 will come. TK for B, 3 will come. Then catalytic perfect enzyme is what? Remaining only one option is there. So triose phosphate isomerase is catalytic perfect enzyme. So option D will be the right option. Clear? So triose phosphate phosphorase is catalytic perfect enzyme. Exokinase is a substrate induced conformation changes responsible for enzyme activity. Aldolase will be there for perfect stereospecificity will be there. Enolase will lead to the synthesis of high energy compound capable of driving ATP synthesis. I hope this is clear for everyone. Okay. Now simple question, glycolysis. Where the glycolysis will happen? Where the glycolysis will happen? In cytoplasm or mitochondria matrix or inner mitochondria or chloroplast? Where the glycolysis will happen? Yes, cytoplasm is the place where the glycolysis will happen. Yes, clear. Next, who is the Director General of ICAR? Who is the Director General of current Director General of ICAR? I already told in our discussion class itself. They may ask director, Director General. They may ask. You just go through once. Who is the present Director General? Who is the present Director General? Students, any guesses? Any guesses? Who is the current Director General of ICAR? Yes. Excellent. Dr. M.L. Jatser. M.L. Jatser is the current DG of ICAR. Yes. Yes. Excellent. Next, about glucose transporter they have given. Glucose transporter they have given. Read the question properly. Which of the following glucose transporter specifically transport fructose? They are saying that is specifically transporting the fructose, and thus that plays a key role in dietary fructose uptake and metabolism. That is playing a key role in dietary fructose uptake and metabolism. Which will be the right option? This is most important thing they have asked in CSR examination also. GLUT5 is specific for fructose molecules. GLUT5 is specific for fructose molecule. This you have to remember. GLUT5. GLUT5, not GLUT1 and 3. GLUT5 is specific for fructose. GLUT5. A typical symptom for calcium deficiency in plant. Typical symptom for calcium deficiency in plant. What will be the typical symptom? Students, what will be the typical symptom? First, if you see in plants, this calcium is what? It's an immobile. Calcium is an immobile element. So, if it's an immobile, the symptom will first happen in the younger leaf. This is clear. Symptom will always happen in younger leaf. Yes, excellent. So, you see older leaves they have given, this will not come. Stem premature leaf fall, this will not come. So, at younger leaf necrosis will be happen due to calcium deficiency. This is another most important thing and most simple question also. Any guesses, students? Any guesses? Different thing. Meristem culture, for what purpose we will use? cDNA library, for what purpose we will use? Lambda phage and cryopreservation. Match the lists and let me know the right answer. Meristem culture, primarily they will used for, yes, meristem culture primarily they will use for producing virus-free plants. Yeah, producing virus-free plants. cDNA library, ultimately mRNA we will convert into cDNA, that will be converted into cDNA library. Lambda phage, we know, Cas site will be there, that is cohesive site will be there. Cryopreservation, DMSO will be used as a chemical in cryopreservant. Dimethyl sulfoxide. This can be used as a cryoprotectant. Another question. See here properly, another question they have asked about enzymology only. Somewhat tricky question. Somewhat tricky question. Which of the following class of protease does not use catalytic triad? They are not using catalytic triad to hydrolyze its protein substrate. So from first sentence we are clear. Chymotrypsin. ठीक है, chymotrypsin. This we can also say as serine protease. ठीक है, serine protease. This will use catalytic triad, but they are saying that does not use catalytic triad. So chymotrypsin will not come, serine protease will not come. ठीक है, I have eliminated this option. ठीक है. Second statement, what they are saying, but rather use one-step catalysis. It is using only one-step catalysis in which an acid is utilized. See here, acid. You see here among them, which will be the acid? Among them, aspartic acid is an acid. Okay, I am keeping this thing aside. Aspartic acid is utilized to water molecule, which then further hydrolyzes the peptide bond in the protein substrate. So we can easily say from here itself, it is an acid protease. Okay, so the right option will be aspartic protease. Okay, example of this acid protease, this they can ask us. Okay, pepsin, renin. Okay, and most important thing is HIV protease. Okay, HIV protease is an example of aspartic acid protease. Okay, clear? So the right option for this is aspartic protease. Okay. Then another question from translation they have asked. Okay. Which among the following facilitate binding of eukaryotic messenger RNA to ribosome? See, eukaryotic mRNA is there. That has to be bind to the ribosome. Okay, which will help this process? Which will help this process? Which will be right? Which will be right? 7-methyl guanosine cap. 7-methyl guanosine. Yes, 7-methyl guanosine cap. See in the here 5' cap structure will be there. This will act as a site where ribosome will bind in eukaryotic. Then in prokaryotic, which will help? Prokaryotic, what will become here? In this option only it's there. For prokaryotic means which sequence will help? There comes the role of Sine-Dahlgren sequence. Okay, Sine-Dahlgren sequence will come there. Okay, what is Sine-Dahlgren? It's a purine-rich sequence. Okay, it's a purine-rich sequence. Okay, so for example, this is the mRNA of prokaryotic. Here, purine-rich sequence will be there. This will be complementary to the ribosome. Okay, this ribosome, we know this ribosome will have RNA molecules also. Okay, that's why we are saying ribonucleotide proteins. Okay, so this will be complementary here, so it will go and bind. So the prokaryotic means Sine-Dahlgren sequence, eukaryotic means 7-methyl guanosine cap. Then another thing, simple definition they have asked, what is osmosis? What is osmosis, transpiration, plasmolysis and cohesion tension theory? Match and let me know the answer. Match and let me know the answer. Yes. Yes. Osmosis is a passive movement of water. This is clear. So C will come option. Here option is wrong or what? You see, transpiration means what? Transpiration means it's a loss of water. So loss of water from these things from leaves will be happen. Plasmolysis means shrinkage of water from the cell. Okay, so C will come option 1. Cohesion tension theory for what? How the water movement under cell they have given. Okay. Okay. So 3, 4, 1, 2 is the right answer. Okay. Clear, students? 4, 3, 1, 2 is the correct answer. 4, 3, 1, 2. Okay. Next thing, in most oil seeds, during germination, the extent of acetyl-CoA used for energy production via respiration is what? Okay, how much percentage will be used? Here you have to remember about the glyoxylate cycle also. Okay, these are some of the important points they can ask. They are asking what is the exact percentage or approximately how much percentage of acetyl-CoA will be directly utilized for energy purpose in respiration. Okay, this is some of the static question. Okay, 30% it will use. Okay, just you remember. Okay, like this question they may ask. Okay, Then see here transamination reactions. This we have discussed clearly in our amino acid metabolism class. They have asking here what are the different substrate or alpha keto acid. If amino group is attached here, what are the acids will be formed? Amino acids will be formed here. Okay, you have to match here. We know oxaloacetate will be a this if we accept amino acid it will become aspartate will be formed. This we know. Alpha keto glutarate means what? Glutamate will form. This also we know. Pyruvate means what? Alanine will form. Hydroxypyruvate means serine will form. Just match this one. So pyruvate means what? Alanine will form. Option this is for 3. Hydroxypyruvate means serine will form. So wherever 3, 4 is coming, 3, 4 is coming, okay. Oxaloacetate means aspartate. This is third one. Alpha ketoglutarate means glutamate. This is fourth one. Okay. So the right option is what? 3, 4, 1, 2. 3, 4, 1 and 2. Option C is the right option. Yes, correct. Next, net assimilation rate. See, morning session also we have seen one class from this crop growth parameter. Now also we are seeing another question. Okay, morning we have seen about leaf area index. Now we are seeing about net assimilation rate. What is meant by net assimilation rate? It's a biomass produced per unit leaf area. Yes, biomass produced per unit leaf area. This is a direct question. Okay. So this is clear. We have to remember what are the different growth parameters and what is the units and who has discovered that parameters and what is the purpose of that different growth parameters. Okay. This you have to remember. So 2 questions they have asked. Okay. Next question. See how they are asking. 5' adenylate acid is chemically and it's simple. This adenylate acid means what? It's an AMP, adenosine monophosphate. Another name is adenylate acid. What will be the answer? Whether it's a nucleoside or nucleotide or nitrogenous base or ATP molecule. What will be the right option, students? Adenylate acid is adenosine monophosphate. So here it will have phosphate group also will be there, base also will be there, and sugar also will be there. So it is an what? It's a nucleotide. Excellent. It's a nucleotide. Next, by convention, general agri question, genetics also we can say. By convention, the short arm of chromosome is called as what? Short arm of chromosome we will call as what? Whether it's a P arm or Q arm. You have to remember like these questions. Okay. Yes, excellent, 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 excellent. Excellent. Okay, P arm. Okay. P arm is what? It's a short arm. Okay. Long is Q arm. Okay. You just write this one. Okay. P arm means short. Long arm means large. Okay. Next. Which of the following class of membrane constitute is sterically similar to glycerophospholipid? They are asking which of the following class of constitute is yes, excellent, sphingomyelin. See, if you see the structure of this glycerophospholipid, glycerol backbone will be there, 3 fatty acid will be attached here, here phosphate will be there with this head group will be there. Okay. They are asking which will be sterically similar to this structure. If you see the sphingomyelin structure, sphingonine backbone will be there, fatty acid will be there, phosphorus and choline will be there. So this will have a similar structure. And during our class itself, I have told this is the only sphingolipid having phosphorus. Okay. So only phosphorus containing sphingolipid is sphingomyelins. Okay. Sphingomyelins. Okay. Next. Who was the first person to develop DNA fingerprinting? Scientists they are asking. This is direct question. Who was the first person to develop fingerprinting, DNA fingerprinting, who has developed that one? This is a static question. Alfred Jeffrey, okay, and it's a direct question also. Alfred Jeffrey is the scientist. Yes, Jeffrey is the scientist. He has developed. Okay. Which of the following enzyme of TCA cycle release carbon dioxide? Observe properly. In TCA cycle, what are the steps where carbon dioxide are releasing? Okay, there will be a two-step. There will be a two-step. There, carbon dioxide will be there released. What are the step? One will be by alpha Kg dehydrogenase. Another is isocitrate dehydrogenase. These are the two steps. So alpha ketoglutarate dehydrogenase and isocitrate dehydrogenase. This is clear. This is a simple question. You will answer. Next, the organelle associated with photorespiration in C3 plants. What are the organelles associated with photorespiration in C3 plants? We know CPM. CPM, this we already know. Chloroplast will come, peroxisome, and mitochondria. These are the organelles involved in C2 cycle or photorespiration. So, among this answer, chloroplast is the right option. Okay, chloroplast is the right options. Okay. Which of the following histone bind to linker DNA? They are asking linker DNA. You have to observe clearly. They are asking which of the following histone will bind to the linker DNA. We know histone octamer will be there. Here DNA will be wrapped. Histone octamer DNA will be wrapped. Here the linker histones will be there. What will be that linker histone? Yes, H1 is the linker histone. Excellent. This octamer will be formed by H2A, H2B, H3 and H4. Okay. H2A, H2B, H3 and H4 will be formed as an octamer. Okay. That is nucleosome complex will be formed. Swiss prot. It's an example of secondary database. See, 3 questions they have asked from bioinformatics. Okay. Three questions they have asked from the tools. So you have to remember properly about the tools. Okay, what are the tools they are used in bioinformatics? Okay, so Swiss prot is one of the primary example for secondary databases. Okay, this is an example of secondary database. Okay. Next, which is the following is viral genome is double standard DNA. See, morning session we have seen. They have asked which of the following is single standard DNA. That is Gemini virus. Here they are asking. Here they are asking double standard DNA. What will be the answer? What will be the answer? Yes, cauliflower mosaic virus. Excellent. Cauliflower mosaic virus. Yes. Next, they are given a different scientist name along with the contribution in photosynthesis. See, if you don't know the answer, just select by elimination method. See, at least one of the things we will be familiar. For example, Priestley, he only concluded oxygen will be released during photosynthesis. Whether it's there, yes, plant release oxygen. So, D will come option 1. You see here, only this option has D as option 1. Okay. So this is the right answer. Then another thing is what? Bossingalt, what he did, he has done nitrogen uptake by plants from the soil and atmosphere. How it is happening like that he told. Yeah, 2314 is correct answer. Julius Van Sachs, what he did? He studied the importance of water culture in plant nutrition. Okay. This is simple question. Okay. So go by elimination method also. If you know some of the things, you just apply that, you just eliminate the option, whatever is not there, then you work out and select the right option. Okay. Next, they are asking about E. coli DNA polymerase 1. This we have already discussed. Which of the following activity is not found in E. coli DNA polymerase 1? Any guesses? Yes, 5' to 3' polymerase activity will be there. This we have discussed. Proofreading activity will be there. Exonuclease activity will be there. Okay. Okay. It has both polymerization and proofreading activity in the same polypeptide. Any guesses? What will be the correct answer? Yes. Yes. Very good, students. Next, which of the following light absorbing pigment is present almost in all oxygenic photosynthetic organism? They are asking, it will be present in almost all oxygen evolving photosynthetic organism. Okay, almost all oxygen evolving photosynthesis organism. So that may be a primary pigment. Okay, primary pigment. Among this, chlorophyll A is directly involved. Okay, directly involved in light reaction. Okay, directly involved in light reaction. Okay. Then if you see, most important thing, this all are accessory pigment that will absorb the light energy, then it will transfer to chlorophyll A. This contents may vary or presence or absence may vary, but chlorophyll A will always present. So the right option is chlorophyll A. See, another important thing about what? Harvest index. They are asking about physiological maturity and harvest index. Read the question properly. Identify the crops wherein the formation of black layer is visible as a spot at the base of the grain as index of physiological maturity. What will be the answer? Yes, option A. Excellent. Option A. Okay. So sorghum is the okay. We can see this one. So sorghum is a typical example. Sorghum is a typical example where the physiological maturity will be done by the induction or formation of black layers. Okay, so the right option is option A. Okay, identify the plant where the stomata are altogether absent or if present they are vestigial. This you have to see. Okay. Yes, log 46, 112 ka B answer. Okay. We will see that one. First you see this. Okay. Identify the plant where stomata are altogether absent or if present they are vestigial. Vestigial. Pondweed. Vestigial. Yes. Potamogeton. That means what? Means these plants that are growing in submerged condition. For example, hydrilla, like that plants will be there. Na? That stomata will be functionless. Okay, that function will not be there. Okay, so this is another important thing. Okay, another important thing. How the stomatal arrangement is there. Just you go through this one. How the stomatal arrangement is there. Just you go through this one. Okay. Next, the indirect synthesis of triacyl glycerol from phospholipid involves the following enzyme. They are asking here from lipid biosynthesis question. Okay. The indirect synthesis of triacyl glycerol from phospholipids involves the following enzyme. Which enzyme will be there? Like what are the enzymes? We know phospholipid will be there. Okay. This is a different classes. Okay. Phosphatic acid will be there. Phosphatic acid. From this phosphatic acid, if the phosphorus is removed, that will lead to formation of diacylglycerol. Here, one more fatty acid will be binded, means that will lead to formation of triacylglycerol. Okay. They are asking indirect synthesis. Okay. The major enzymes what? Choline phosphotransferase, diacylglycerol acyltransferase. This is right. We have seen now. Yes. Then phospholipid diacylglycerol acyltransferase, transferring the acyl group from phospholipid diacylglycerol. So option will be A and B. Okay. Then direct questions only they have asked here. See here, scientist contribution they have asked. First action spectra. First action spectra in photosynthesis using an aquatic algal filament and oxygen seeking bacteria was given by which scientist? This they have given in BK Jain. During our class itself also they have told during the photosynthesis class itself. Okay. Angelman, he is the person who has first used this action spectrum and he is the first person who has discovered these things. Okay, this is most important thing you have to remember. Okay, Von Neil, what he did? Oxygen is evolving from water and not from carbon dioxide. During photosynthesis, oxygen will be evolved That oxygen is coming from water only and not from carbon dioxide. This was given by Van Neel. Robert Emerson, what he did? Ruddrop and Emerson enhancement effect. Ruddrop and Emerson enhancement effect was given by Robert Emerson. Okay. Arnold, what he did? Criteria for essentiality of nutrients. Okay. You just remember criteria for essentiality of nutrients. Okay. Then another thing they have given about hemoglobin. Okay. How the hemoglobin is inhibited. Yeah, criteria. Yeah. How the hemoglobin is inhibited. Okay. What are the different subunit? What is the difference between adult hemoglobin and normal? This adult and fetal hemoglobin. Before going this, we have to first remember 2,3-bisphosphoglycerate is a inhibitor. That will competitively bind to the oxygen, so that oxygen binding capacity will be inhibited in hemoglobin. Then another most important thing, this fetal hemoglobin will have different subunit. If normal hemoglobin means alpha 2 beta 2 will be there, here the thing will have gamma 2 subunit. Alpha 2 and gamma 2 subunit will be there. This will have less affinity for this 2,3-bisphosphoglycerate. So 2,3-bisphosphoglycerate will be less efficiently bind to this gamma subunit because of this oxygen binding capability of this fetal hemoglobin will be higher. Okay, you read the question now. The fetal hemoglobin has greater oxygen affinity. This is true. Then adult hemoglobin, correct? Because of its higher binding with 2,3-bisphosphoglyceraldehyde. They are saying this 2,3-bisphosphoglyceraldehyde. See, this 2,3-bisphosphoglyceraldehyde is binding because of this higher affinity is there, like that they are saying. So this is wrong statement. If 2,3-bisphosphoglycerate will bind means that will decrease the affinity. Then second thing, reason, the BGP binds less tightly to fetal deoxyhemoglobin. This is true. Then adult deoxyhemoglobin. So only R is correct. R is correct, but A is false. Students, then one more thing I want to say. Whatever we are discussing here now, this is the right options. Maybe some of the mistakes will be there while typographic errors. In the answer key, if anything mistake is there means we will solve there only. Don't be confused. Typographical errors may be there. Next simple question about alpha helix they have asked. We know alpha helix, about alpha helix structure they have asked. The distance between each amino acid in alpha helix is what? 1.5 Armstrong will be their distance. This is static question. We know 1.5 Armstrong will be there. Assertion what they are asking, in globular proteins, alpha helix has an average span of 12. They are saying 12 amino acid is there. And then average length of 18 Armstrong. See, 12 Armstrong means 12 into 1.5 Armstrong. So total 18 Armstrong will come. So assertion is right only, correct? Assertion is right. Reason, the rise per amino acid residue in alpha helix is 1.5 Armstrong. This also they have given correct only. This we already see. So both A and R are correct, and R is the correct explanation of A. This is clear. This is clear. Yes. Next, last question. Identify the amides, primarily amino acid asparagine and glutamine, exporting legumes. They are asking here, what are the amides? Amide exporting legumes. This during our nitrogen metabolism itself I have told. Amide and urides. Urides exporter will be majorly pulses like soybean. This is the major uride exporter. Major uride exporter is soybean. Apart from that, groundnut oilseed, groundnut also will export the fixed ammonia. Fixed ammonia into uride. So what will be the answer? What will be the answer? Soybean also will come. Then groundnut also will come. Whether any options is there? Yes. A and C. But clover and lentil is a typical example for amide exporter. main exporter. Clear, students? Clear? Any doubt is there? Any doubt, students? Kindly let me know, any doubt is there? Any doubt is there? Kindly let me know. Yes, 112 में official answer की according to B. 112. Okay. Answer. Lok. Answer. Yeah, Uday Lakshmi have no doubt. Okay. Lok 46. 112 में official answer की according B है. You just tell. Polymerase activity will be there. Yes, 5' to 3', polymerase activity will be there. This is true. 3' to 5' exonuclease activity will be there. This is also true. Correct. 5' to 3' proofreading. We have to read the question properly. Here they have given proofreading. Whether that is a proofreading? Any guesses, students? It's a not proofreading. It's an 5' to 3' exonuclease. It's a 5' to 3' exonuclease activity will be there. We can also say this is as primer removal activity. Primer removal activity. Look, excellent. Okay. Excellent. So this is the right option. Okay. 5' to 3' proofreading. You have to read the options also properly. Okay. 5' to 3' proofreading activity will be there. Okay. Students, then one more thing, kindly comment if you have any doubt is there means. Okay. If this session is also useful means kindly let us know. We will continue with this section. Okay. Thank you, students. I will see you later in next classes. Okay. Thank you for your interactions. Thank you. You may leave now. Thank you. Not yet.